All right, so phosphorylation, dephosphorylation, we added a highly charged phosphate group to a neutral amino acid in serine, threonine, or tyrosine, and that caused hydrostatic or van der Waals forces, which, if it's a negative charge, it's going to pull towards a positive, and it's going to push away from a highly negatively charged molecule. By contrast, acetylation, deacetylation, what we're going to do is we're going to add an acetyl group. An acetyl group is this group right here, and what you can see is there's no charge. So it's a neutral group, and what we're going to do is we're going to add it to a lysine. And remember, a lysine is a positively charged amino acid. So when we add an acetyl group to a lysine, what we do is we take a positive charge and we turn it into a neutral. So we remove positive, negative, strong interaction from those amino acids. These enzymes that are going to do this are called acetyltransferases. They transfer acetyl groups, and what they do is they take the acetyl group off of acetyl-CoA, what we made through glycolysis or we made through beta oxidation. They're going to take that acetyl-CoA and they're going to take the acetyl group off of it and they're going to add it to a lysine amino acid. The enzyme that takes it off is just called a deacetylate. It removes the acetyl groups. And again, what the acetyl group does is it changes the charge of the lysine molecule from positive to neutral. And by doing that, what it's going to do is it's going to change the shape, the activity, the binding proteins or the binding partners, or the location of that protein. So really, acetylation, deacetylation has the exact same four functions as phosphorylation, dephosphorylation. You're going to change the charge of the molecule, and that's going to cause it to either become more active, it's going to change the shape of the protein, which is then going to potentially change its binding partners and its location within the cell. And again, this is something that happens all the time when we talk about exercise, and we're going to talk about nutrition. Examples of acetyltransferases and deacetylases. The deacetylases are the sirtuins like SIRT1 and SIRT3, which are deacetylases that are activated by NAD. This is happening when we're doing lots of endurance type exercise. And histone deacetylase itself, again, that's deacetylating the histones, and that's going to cause the DNA to change its openness. And when that happens, this is something that's going to be key to the endurance adaptation. And when we look at the other side, the acetyltransferases, we're talking about things like GCN5, which is activated by exercise, P300 and CREB binding proteins. These are other forms of acetyltransferases that can be activated by diet and exercise. And we're going to take advantage of those things because those are important in the adaptation to exercise and diet. So how does this work? You're going to take this, again, our weird looking protein here that has a lysine. It's a positively charged group, as we said before, and it's here interacting with negative charges. Think about this. What is a negatively charged molecule within the cell? DNA. DNA is negatively charged. So when you have acetylated proteins, they can maybe change their ability to interact with DNA. And that's, again, when you acetylate, deacetylate histones, that's why it opens and closes DNA. All right, so when an acetyltransferase takes the acetyl group off of acetyl-CoA and transfers it to a lysine, that's going to make this into a neutral, and that's going to decrease the affinity for this negatively charged molecule. In the reverse, the deacetylase is going to remove that, and it's going to allow that interaction to happen again. And again, that's going to change the shape, location, activity, or binding partner of this protein. All right, so acetylation, deacetylation, very similar to protein phosphorylation, dephosphorylation. Somewhat different now is we're going to talk about these things that are called agonist binders. And agonist binders are basically, it's the binding of an enzyme or transcription factor or some other protein to its substrate. And what that can do is that can change the activity of that protein. Some of the best examples of this are small GTPases. This is going to be really important when we talk about how leucine-rich protein is activating or increasing muscle mass, because all we're doing with leucine-rich protein or even with resistance exercise is we're changing the activity of a series of small GTPases that are, most of them, when they bind GTPA, they're active. And then when they hydrolyze the GTP to GDP, they become inactivated. So it's like a time-dependent structure. So it's like it's a structure that turns itself off in a certain amount of time. It binds GTP and it very slowly is going to cut it off itself. But while it's still got the GTP there, it's highly active to do something within the cell. When it finally cuts off the GTP, it's like your egg timer has gone off, the enzyme is turned off again, and it needs to be reset. So you can also have agonist binding to activate transcription factors. One of them that we're going to talk about is this one called the PPARs. And the PPARs are a series of fatty acid activated transcription factors. They directly bind fatty acids, and when they're bound to specific fatty acids, they have higher activity. And these enzymes are going to be important for making all of the enzymes that metabolize fats. So when you have more fats in your muscle, you're going to turn on the PPARs. That's going to turn on your ability to break down fatty acids. Another example of an enzyme now that's activated when it binds its agonist is glycogen synthase. If you're going to make glycogen, the enzyme glycogen synthase can be directly activated by binding to glucose. When it binds glucose 6-phosphate, it becomes activated and it increases the rate at which it synthesizes glycogen. So in all of these different ways, we can, just by binding a protein to something that it's supposed to be interacting with, we can actually directly increase its activity. All right, so examples of this. This is how a G protein works. When it's in the off state, it only has GDP here. You can see there's 2 phosphates on this guanine. And then what you need to do is you need to pop that out, because that's very tightly bound in there. And this binding is actually very strong. It's stronger than it binds to GTP. And so what you need is what's called a guanine nucleotide exchange factor, or a GEF protein. And what a GEF protein does is it pops the GDP out of the small GTPase. And when that happens, there's 10 times more GTP in the cell than there is GDP. So if I pull out the GDP and the GTP is in the cell at 10 times higher concentration, the likelihood of it binding to a GTP is much, much higher, and that's going to turn this protein on. If I want to accelerate the egg timer because I set my timer, I set it for too long, now I'm going to turn it back and shorten the length of time of my timer, you can do that using a GTPase activating protein. And what these are GAP proteins, and what they do is they accelerate the rate at which this GTP gets cut into GDP and you release inorganic phosphate. So I can turn on a small GTPase by using a guanine nucleotide exchange factor. I can turn it off using a GAP protein. We're going to see this when we talk about how amino acids increase mTOR activity and muscle protein synthesis, because amino acids activate or alter this GEF protein on this small GTPase called the RAGs. And when we get it active, that RAG can now increase things that are going to drive protein synthesis. When we fast, we activate a GAP protein that's going to turn this whole system off so that we don't increase protein synthesis when we're fasting. If we look at the PPARs, they're a transcription factor. When a fatty acid comes in, it changes the shape of this protein because as it binds, it grips onto it. And then what it can do now is it can bind better to DNA. So when it's bound to the specific fatty acid that activates it, it can now change its shape, it can bind to the DNA, and it can regulate gene expression.